Hello, everyone. My name is Edwin Appiah, and I'm from Kwame Nkrumah University of Science and Technology, Ghana. And this afternoon, I'll be presenting to you the Gastric Microbiome and Gastric Carcinogenesis, with much focus on bacterial diversity, co-occurrence patterns, and predictive models. So this is going to be the outline of my presentation. So gastric cancer is the fifth most common cancer in the world and the fourth leading cause of cancer-related death, according to the Bakan report in 2020. It contributes about 5.6% of the total cancer-related incidences and also 7.7% of the total cancer-related deaths. Gastric cancer is predominantly caused by your lifestyle, what you eat, what you drink, high smoking, alcohol. But most importantly, there are also some health-related factors here, like your blood group, your genetic predisposition, and microbial infections, which has been established that Helicobacter pylori is the #1 carcinogenic factor for gastric cancer. However, only 1 to 3% of people who get gastric cancer actually, who are infected with Helicobacter pylori pathogen actually get gastric cancer. And it has also been reported that people who have been, after the eradication of the pathogen, people still go ahead to get gastric cancer. And the gastric microbiome has been thought to contribute to gastric cancer development. However, its role is still unclear. Typically, focusing on intestinal gastric cancer, which is the most cancer that is affected by Helicobacter pylori, we realize that as carcinogenesis happens, moving from atrophic gastritis to intestinal metaplasia to gastric cancer, H. pylori suicide actually happens. And it has been established that other microbial communities could actually come into play or could come in to actually cause gastric cancer. So it has become important for us to try and look at these microorganisms and how they function. This will help provide insight into developing potential prevention and treatment strategies. So many studies have tried to answer some of these questions to look at how these microorganisms happen, but the microbiome typically is affected by your diet and your geographical region. So this has mainly resulted in inconsistent results. So it became important that we try to pull these data sets together and try to look at, irrespective of your geographical region or your diet, if you're able to control these factors, what kind of microbial communities are we going to observe and how are the microbial communities going to interact? That was what we thought out to do in this particular study. So we combined 985 samples across various studies to look at the microbial diversity and composition, the interaction between these microorganisms, and ultimately try and identify microbial biomarkers for gastric cancer carcinogenesis. So this was the methodology. We employed 6 different studies across 985 samples. We used the DADA2 workflow. We controlled for batch effects, that is the location and geographical region, and then also for the 16S method that was used, the 16S hypervariable region that was also used, and then also our co-occurrence patterns were used. We used spring and Briquettes distances, and then we employed lasso for our predictive models. So looking at the Shannon diversity, we realized that as carcinogenesis happens, the diversity actually reduces, which is contrary to what has been reported earlier on by some studies. So, however, this diversity changes, or how the diversity across the various was also significant. Looking at the beta diversity, even though we can't see a clear-cut distinction between the various conditions that we employed in a particular study, which is the cancer, gastritis, healthy control, and metaplasia, the Permonova test that we performed also was significant, which gives us an indication that different microorganisms could actually be involved in gastric carcinogenesis. But we try to do some exploratory studies and look at the microbial composition. And then we found out that 6 phyla actually dominated the gastric microbiome. That's the acidobacteria, actinobacteria, firmicutes, the fusobacteria, and then the proteobacteria. We look at the relative abundance trend of these phyla, and then we found out that actinobacteria, for instance, increases with parthenogenesis, which we could see in a clear-cut distinction here. And then also proteobacteria in general also decreases with carcinogenesis. And it's also worth noting that proteobacteria contains bacteria like Helicobacter pylori, which has already been established that as carcinogenesis happens, its diversity and its dominance reduces in the stomach. But we try to establish the microbial interaction between these microbial communities, and then we also try to look at the interaction and which of them co-occur here. So here, generally, we in the healthy control individuals, 5 clusters were obtained, and then which is color coded. So each of the colors represents a cluster. The node sides also represent the abundance, and the hubs are also have higher node sizes. So we realized that Sphingobiome's phagum was actually represented as a hub, together with Zuglia was philia, which is also represented as a hub. And this gives us an indication that these microorganisms could actually be controlling the healthy individual, the microbiome of the healthy individuals. We look at the gastritis, and then we found out that Valiona despair, and then, but most importantly, we realized that Prevotella species, which have been reported in gastric, which have been reported in inflammation related diseases, also increased as carcinogenesis also increased. And then most of these bacteria actually interacted with several other bacteria. I would like to focus on the gastric cancer, or the interaction between the within the gastric cancer community. And then we found out that the Prevotella species actually co-occur with some, and notably some carcinogenic species like Fusobacterium nucleatum, which has been reported to actually cause or to be involved in colorectal cancer in mouse models. So it gives us an indication that these species, or these transient species, actually come in a later stage to interact to cause gastric cancer, which we didn't see in our previous interaction when we compared it with the healthy control individuals. And then also Filifactor allosis also co-occur with Fusobacterium, and these co-occurrence, these bacteria actually depend on each other for survival, which gives us an indication that probably Filifactor allosis, which is represented as a hub in the gastric cancer conditions, might actually be there to facilitate the growth of certain carcinogenic bacteria like Fusobacterium, for instance, as indicated in this particular study. But we now try to look at the core microbiome and how they interact. And interestingly, when we pick the same microbial communities in a healthy individual and then in a cancer individual, it turns out that their interaction differs, and their co-occurring patterns also differs. And for here, we realize that Cartonella morbi is represented as a major hub, which is driving the healthy control community. But when we come to the cancer community, it is not represented as a hub. Likewise, we see for the Bacteroides species here. So in the predictive model, we tried to build a lasso predictive model, and then we had a mean AUC of 0.83%, 0.9, which is quite high. But we are quite skeptical, so we are trying to fine-tune this particular model, but it's also noting that Bacteroides dori, which is represented as a herb, when we did a co-microbiome comparison with, we have some Bacteroides dori, which is also with a robustness of 100%, giving us an indication that Bacteroides species could actually play a key role in gastric cancer carcinogenesis, because we are seeing them represented as herbs. And then we also see in our predictive model, we have Bacteroides dori being selected as a discriminatory factor for healthy control individuals and then gastric cancer. So we believe that these species might actually play a key role in the pathogenesis of gastric cancer. At the end of the day, microbial diversity and composition differed significantly across the various histological stages. We realized that transient oral bacterium also dominated the gastric microbiome of gastric cancer individuals. And then most Prevotella species also co-occurred with Fusobacterium nucleatum. Bacteroides dori was actually identified by a predictive model to be a potential microbial signature for gastric cancer. Currently, we are trying to validate these results using African population. So we are collecting samples at the moment, where we are going to do a 16S to try and validate these results. So I would like to acknowledge my supervisor, Dr. Samson Pandam Talifu. I would also like to acknowledge Waslibre, KNUST, and the National Institute of Mathematics. Thank you very much.